Dear colleagues, today we will talk about metabiotics as a new step in the evolution of biotics. And we will talk about what are the metabiotics, how are they classified, what are their mechanisms of action, and what are their potential therapeutic uses. We all know that human microbiome consists of trillions of cells. And the biggest microbial consortium is in the human gut. It is the main microbial community that is determining all the other microbial communities in the organism. And each person has its own unique intrinsic microbiome. Microbes in the gut produce microbial factors. So these are, for example, the structural molecules like the different molecules of the outer membrane, outer cell wall of bacteria, like teichoic acids, peptidoglycans, lipopolysaccharides, genomic DNA also, and a number of intracellular and extracellular metabolites like bile acids, fatty acids, indoles, vitamins, amino acids, antimicrobial factors, also neurotransmitters, and so on. And these substances have a local effect in the gut, like, for example, immunomodulation. They support the normal microbiota, they fight pathogens, and also they support the normal function of the epithelial layer. And at the same time, they also have systemic action, like anti-inflammatory, anti-neoplastic, anti-mutagenic, and even anti-hypertensive. So how does it happen that they have a systemic action? The fact is that the low molecular weight metabolites and also some structural elements produced by microorganisms, for example, by bacteria, they can surpass the epithelial layer. They can go through the subepithelial tissue and end up in the blood flow and also in the lymph flow. And by this, they can reach the peripheral tissues and organs and exert a systemic action. So in this way, a gut microbiome is connected with virtually all the systems of the human body. So we are talking about the so-called interconnection axis between a gut microbiome and the organs like brain, heart, lung, liver, pancreas, and so on. And in case of eubiosis, normal state of gut microbiota, all these systems function normally. But if it comes to the disbalance of microbiota, dysbiosis, that can be caused by various negative factors like wrong lifestyle, wrong diet, pollution, drugs like antibiotics, hormones, and so on. And the result of this disbalance of microbiota will cause various illnesses. So therefore, since the middle of the last century, the so-called biotics have been used for the correction of microbiome. And the most well-known are probiotics. These are the living cells of beneficial microorganisms, mostly bacteria that are used. Then we also know prebiotics. These are the substrates for the beneficial microorganisms, and symbiotics are the combination of the first two variants. Probiotics are widely used, but they have their concerns. They need time to start acting because the cells need to revive in the gut. They need to start metabolizing and propagating. Also, they are vulnerable towards the inner and outer environmental factors, like temperature or bile acids, for example. They are also sensitive towards antibiotics if they don't harbor specific antibiotic resistance genes. And they have safety concerns. So, for example, the International Association for Probiotics and Prebiotics has published last year a consensus paper on the safety issues with probiotics. And they have listed the following issues that, first of all, it is widely known that probiotics, if they are carrying the antibiotic resistant genes, they can transfer them over to other microbes in the organism. They can also cause bacteremias in the vulnerable patient groups like older people, preterm infants, immunocompromised patients. They can interact with drugs with unpredictable results. They also may have negative adverse effects, or they may have no effect at all, depending on the intrinsic microbiota of a certain person. And also, it has been noted that they can even interfere with the normal microbiome of the person. And that's why the interest towards a new type of biotics has raised. So a new generation of biotics has been raised in the 21st century. And these are the microbial-derived substances that do not contain living cells. So, for example, metabiotics. So this term has been first introduced by Russian scientist Boris Shenderov back in 2013. And today we understand under metabiotics the whole range of bacterial metabolites and signaling molecules like fatty acids, antimicrobial substances, amino acids, enzymes, vitamins, lactic acid and its derivatives, and so on. Also the structural molecules, like the molecules of the cellular wall, like peptidoglycans, polysaccharides, teichoic acids, also nucleic acids, and the whole inactivated, non-viable bacterial cells also belong to this class of substances. So if we look in the literature, then we can see that along with metabiotics, we can find also postbiotics, paraprobiotics, parapsychobiotics, ghost biotics, Tinderlized probiotics. And actually all these terms mean the same thing. So these are bacterial-derived substances that do not contain living bacterial cells. So bacterial lysates will also refer to this group of substances. And if we look at the PubMed database for the publications with these substances, we can see that really the last years have seen an explosion in the number of publications about metabiotics and similar substances. So what makes them attractive? Well, first of all, this is their faster effect. As long as these are the isolated molecules, they start acting faster than in comparison to prebiotics. They are resistant towards the negative factors of the environment. They are also not affected by antibiotics, and they have a higher safety profile. They don't transfer antibiotic-resistant genes. They are not harmful to the vulnerable patient groups, but at the same time, they retain most of the positive effects of probiotics. What is important, they do not interfere with the unique microbiome of each person, but they support this unique microbiome. And at the same time, as long as these are the isolated biologically active molecules, they open a wide possibilities for combining them with other biological substances like prebiotics or plant extracts, vitamins, microelements, for creating formulations that could be targeted towards specific organ systems. So what are the mechanisms of action of metabiotics? So first of all, these are the local actions, combating pathogens. Inactivated bacterial cells can prevent the pathogen biofilm formation. Metabolites contain antimicrobial substances. And also, inactivated cells can physically bind to the pathogens and block their propagation. And this all leads to elimination of pathogens. At the same time, the metabolites contain the substances that are supportive for the normal symbiotic microflora. Also, the local action is on the epithelia of the gut. The metabiotics promote the expression of tight junction proteins. They also support the mucus production, that is a defending layer for the epithelia. And what is also important, they switch on the anti-inflammatory cellular processes. This can happen as a result of immunomodulatory action of bacterial substances. So, for example, the structural elements like peptidoglycans, teichoic acids, surface layer proteins, and so on, also the genomic DNA, can interact with the specific receptors on the dendritic cells. These are immune cells in the intestine. And this interaction switches on the downstream cellular cascade that leads to the production of cytokines that can then signal to the T cells. Also, the metabolites can act on the dendritic cells and on the T cells. And as we remember, as long as the low molecular weight substances can suppress the epithelial layer, then it means that metabiotics can also come into the blood flow, into the lymph as well, and there they can interact with the immune cells in the peripheral organs, and by this they can lead to the systemic therapeutic effects. So what do we know about the studies on metabiotics? So, for example, the review from last year has listed 24 randomized controlled clinical studies on metabiotics and similar substances that were found in the Cochrane library and in Medline database. So here in the table, we just list some of them. So they are mainly, these are on bacterial lysates and on the inactivated bacterial cells of various species and strains. And here we can see the effects that they show positive results in the irritable bowel syndrome, the menopause symptoms, Helicobacter pylori infection, and also atopic dermatitis. So we can see a really wide plethora of effects. So metabiotics are also making their way to the clinical guidelines. For example, the last Maastricht 6 Florence guidelines on the management of Helicobacter pylori infection state that inactivated cells of Lactobacillus reuteri strain DSM 17648 are supportive for the microbiome restoration after the antibiotic treatment, and also reduce the patient complaints over the abdominal pain. So currently the studies on the metabiotics are going in various directions. They are studied for various conditions, for example, like digestive illnesses, Helicobacter pylori infection, irritable bowel syndrome, and others. Infectious disease and their consequences, like, for example, COVID and post-COVID syndrome, liver disease, metabolic disease, like diabetes of the second type, or obesity, respiratory infections, and nervous system illnesses. So to conclude, we can say that metabiotics are a new approach to digestive health and the health of our organism as whole. And also metabiotics create possibilities for effective, targeted, and importantly, personalized interventions that can improve various aspects of human health. And thank you very much for your attention.